I think we should start. Our first speaker in this session is Syed Yusuf Mian from the Jawaharlal Nehru University in India. So please unmute yourself, and once you see your slide, you can go ahead.
Thank you very much. First of all, I would like to thank the organizers for giving me the opportunity to present my work before you all. And today I'll present one of my PhD works. Next slide, please. Okay. So the focus of my research has been to study the red cell invasion process by Plasmodium with an aim to develop a blood stage malaria vaccine. Next animation, please. However, there are numerous challenges against developing a vaccine targeting the blood stages, which involves the high complexity of this process. It's a multi-step process, and there are diverse ligand-receptor interactions that allow the parasites to invade through redundant invasion pathways, and more importantly, a high antigenic polymorphism in the parasite ligands that results in strain-specific responses. Next animation, please. So with these challenges in mind, the focus of this study was to identify essential and conserved antigen targets that elicits potent strain-transcending parasite neutralizing antibodies, and then those antigens can be combined into a multi-component vaccine. Next slide, please. So for this study, we screen parasite neutralizing efficacy of antibodies against a panel of 7 antigens, as listed here. And one of the antigen, as you can see, MSP fusion at the bottom, is actually a chimera of 2 antigens, MSP3 and MSP1. Next animation, please. So antibodies were raised against these antigens, and specifically against the green regions as highlighted in these cartoons. And we tested them again in vitro growth inhibition assays, in which this figure shows percent invasion inhibition against a parasite clone 3D7. And as you can see here, among all the 7 antibodies, the one targeting MSP fusion, CYRP, and RS5 were the most potent, with an inhibition range ranging from around 80% to 90% at 10 mg per ml concentration. The next slide, please. So we then combined these antibodies together in a triple antibody combination and tested them against 5 parasite clones, 3D7, DD2, 7G8, HB3 in Mellencamp at 3 concentrations, 0.5, 1, and 2 mg per ml. And then this figure shows the inhibition at 1 mg per ml for the 10 best antibody combinations. As you can see here, at individual antibody, the highest inhibition that we observed was around 40%. However, as we combine these antibodies in a triple combination, we observed highly enhanced levels of inhibition. Next animation, please. And among all the combinations, the combination of antibodies comprising of MSP fusion, CYRP, and RS5 was the most potent. Next slide, please. Next animation. And as we also found that antibodies against RS5, CYRP, and MSP fusion were acting in a synergistic manner. Next slide, please. So we then tested the efficacy of this antibody combination against P. falciparum clinical isolates that were obtained from India and Africa and were found to exhibit diverse invasion phenotypes. And we selected 12 isolates, 6 from India and 6 from Africa, as highlighted in the bottom table in bold and underline. Next animation, please. So as you can see, our antibody combination of MSP fusion, CYRP, RS5 was highly potent and inhibited the invasion process by all the 12 isolates in a strain-transcending manner, achieving an inhibition of more than 95% at 2 mg per ml concentration, suggesting the highly efficacious and robust nature of this antibody combination. Next slide, please. Finally, we tested polymorphism in these antigens and encompassing in 200 isolates, encompassing 3 continents and 19 countries. Next animation. As you can see here, we observed only 6 SNPs in RS5, 5 SNPs in MSP1, and only a single SNP in CYRPA, suggesting the highly conserved nature of these 3 antigens. Next slide. So, in summary, we have identified a novel antigen combination of 3 antigens, MSP fusion, CYRP, RS5, that elicits potent parasite neutralizing antibodies, and thus established a proof of principle for the development of this antigen combination as a next generation multi-component blood stage malaria vaccine. Next animation. And this study has been recently published in the Journal of Infectious Diseases. Next slide, please. And these are all the contributors to this study. And thank you for listening. Happy to take questions.
Thank you for that very nice presentation. If there are any questions, please put them in the chat box and I can read them. Well, actually, I have a question to start with. So
Yes.
how are you with this antibody combination that you identified now? How do you want to move on? What will be your next step in the lab?
So this antigen, these we have tested with CFA. That's not adjuvant, that is CFA. That is fruits adjuvant. Now we are moving ahead with human compatible adjuvants. So that's the first step that we are moving ahead with these antigens. Once we have identified human compatible adjuvant that gives the most potent inhibition, we'll take that forward in our clinical studies. And we have also optimized the process for the process development of these antigens at large scale. So that has been done. We are moving ahead with our human compatible adjuvant studies.
Okay.
And then we'll take forward that for clinical settings.
Thank you. So there are a few questions in the chat. One is, are you planning to use viral vector for human immunization with this multi-antigen vaccine?
No, we actually, we make recombinant proteins in E. coli, and then we make formulation with adjuvants and immunize it, not with viral vectors. So that's how we do it.
Okay, and another question is, how about the mutation in the RH5 gene? Are those functionally significant?
Yes, some of the mutations have been reported to be functional, and few of them have been reported to actually define tropism. They define the specificity for a specific red cell. For example, invasion has been observed in red blood cells. So single mutation has been observed to be defining that pathway. But importantly, in our case, antibodies against RH5 do not get affected by those SNPs, and we have tested them. So even parasites having these SNPs, they do not get affected by the mutation, and antibodies they inhibit the invasion in a strain transgenic manner without any effect from the SNPs.
Well, okay, thank you again for a really nice talk. We have to move on to our next speaker of today. So this will be Hazel Beverly Gwarinda from the University of Pretoria in South Africa. So once you see your first slide, please unmute yourself and go ahead.
Thank you, Cecilia, for the introduction, and to the conference organizers for this opportunity to share my work as part of my PhD study jointly performed between the Burkholz Lab at the University of Pretoria in South Africa and the Greenhouse Lab at the USCSF. Although large parts of Africa are still heavily burdened by malaria, parts of the Southern African region and indeed South Africa are in pre-elimination. However, despite advanced and well-controlled vector control strategies, South Africa and other countries in the region have not been able to reach their elimination goals at the proposed target dates. While several factors have been associated with continued residual malaria in the region, the contribution of the malaria parasite itself has only recently been investigated in neighboring countries like Namibia and Eswatini. Prior evidence suggests that in high transmission settings, parasite genetic diversity is high, whilst this typically decreases with a decline in malaria transmission. However, transmission hotspots may be maintained through locally diverse parasites. We postulated that this is also true for the northern border regions of South Africa. We therefore aimed to evaluate if parasite genetic diversity contributes to and fuels the sustained and residual malaria in the transmission hotspot in South Africa. Next slide, please. To address this, we generated parasite genetic data by genotyping 26 unique microsatellite loci for parasites isolated from almost 1,200 RDT positive symptomatic malaria cases over a 3-year period. 2/3 of the sample set originated from polyclonal infections, with individuals carrying anywhere from 2 to 6 distinct multiple parasite clones, with a mean multiplicity of infection of 2 observed. This indicates that within host diversity is highly complex. This conclusion positively correlated with the level of outbreeding observed and highly interbred parasites observed in 33% of the samples. The high genetic diversity was reflected on a population level as well, with a high allelic richness observed, and almost all the samples containing unique haplotypes supporting high levels of outcrossing between parasites. Anywhere from 3 to 26 unique alleles were identified per genotype locus, and this supported an overall high heterozygosity within the samples. Next slide, please. We subsequently interrogated causation behind this genetic diversity and questioned the influence of geospatial, temporal, and seasonal variances on the multiplicity of infection. Stratifying the data according to sampling sites could not associate any significant variance to the multiplicity of infection for the majority of the samples. Low differentiation between all sites as described by FST implies free gene flow and possible parasite mixing between these sites. Patient travel history data indicated that 99% of cases were locally acquired, and therefore the genetic diversity could not be associated with imported cases. Additionally, the multiplicity of infection distribution pattern remained stable over time, indicating that the parasite genetic diversity is maintained irrespective of temporal influences. Moreover, the multiplicity of infection was persistently complex throughout the year, with no significant difference in parasite genetic complexity observed between the wet and dry season, suggesting the maintenance of complex infections in spite of vector control implementation during wet season and possible co-transmission of strains. Next slide, please. Taken all together, the parasite population in a malaria hotspot in South Africa is complex and diverse, which is consistent with its classification as a high transmission setting within South Africa. We therefore conclude that parasite genetic diversity is a contributing factor to sustained residual malaria transmission in this area. This is distinct from other low transmission settings in the country, as we indicated in a comparative study, and this heterogeneity in transmission settings and parasite populations will therefore have significant implications for malaria control strategies in South Africa and will require differentiation in our approaches to reach our malaria elimination goals. I thank you all for your attention, and I acknowledge all volunteer participants who made this study possible. Thank you.
Thank you, Hazel. Perfect on time. Questions for Hazel, please put them in the chat.
So Ada was very fast, or that went well?
No, I never interrupted you at 3 minutes because I knew you had only one slide left. So, well, I can ask a question. So do you have any suggestions, like any specific suggestions on how this data could be used for intervention strategies?
Well, because we're only starting to look at the genomic surveillance of the parasite in the country, I would say that genotyping of the samples could be used as an additional surveillance tools, so that at least we are able to track the genetic diversity of the parasite and see whether that is decreasing with the decreasing transmission intensity.
Okay, thank you.
Thanks, Cecilia.
So there is still time for one more question if we have one. If not, I want to thank you again, Hazel, and move on to our next speaker. We're on a tight schedule here. So next up is Nicole Page from Imperial College London in the UK. Once you see your starting slide, you can go ahead, please.
Hi. So I'm a PhD student at Imperial College London, and I'm just going to be presenting today a brief overview of my project, which is investigating the role played by the double sex gene in tissue dimorphisms in Anopheles gambia. So if we go to the first slide. As a brief background to the project, Drew nicely introduced the double sex gene this morning, which is a highly conserved gene in the sex determination pathway in insects. And in Anopheles gambia mosquitoes, this gene encodes two sex-specific transcripts, a male and a female transcript, where in the female, alternative splicing retains this extra exon 5. And in this study done in 2018, they found that disruption of this female-specific exon actually resulted in this intersex pseudo-male phenotype in genetic females. So as you can see in the diagram, these females develop male-like morphologies, most importantly, this modified proboscis, which makes them unable to bite and therefore sterile. And so with based on the fact that females are the only ones that interact with the parasite, we wanted to further investigate the role of this gene in sexually dimorphic sites of parasite interaction. So if we go to the next slide. So the way we aim to do this is to again interrupt this female-specific exon 5, but we wanted to limit this disruption to the midgut, which is one of the main sites of parasite interaction. And so if we look at the diagram on the right, our design was to have a tissue-specific promoter to drive a Cas9 alongside this guide RNA targeting the female-specific exon. And the idea was to insert this out of locus so that the Cas9 would continually cleave this female-specific exon and cause mutations at the target site and therefore disrupt gene function. And so the gene we chose was this chitinase 8 gene. And as you can see on the image on the left, the GFP we used to tag the Cas9 shows the expression in the midgut, and it was mainly localized to the posterior region of the midgut. So to confirm that the Cas9 was active and cutting and creating the mutations in the midgut, we did amplicon sequencing of the target site with pooled midgut samples. And this showed an average mutation rate of 13% mutation in whole midguts. And as you can see from the amplicon sequencing results, the most common mutations we found were all deletions at the target site. So if we go to the next slide. And so the next thing we wanted to look at was if these mutations were actually having any effect on Plasmodium infection and fertility. So on this slide, I'm going to be talking about the 3 samples we tested. WT refers to wild type mosquitoes, which carried no genetic modification. Het refers to heterozygous for our transgene, so 1 copy of Cas9 and guide RNA, and HOM refers to homozygote for the transgene, so they carry 2 copies. And as you can see on the graph on the left, when we looked at infection with Plasmodium burgii, we compared the number of oocysts that developed on individual midguts, and we saw a significant reduction in infection in individuals carrying these disruptions of double sex in the midgut, and even more so in individuals that carried 2 copies of the Cas9. And so it seemed like this double dose of Cas9 resulted in more cleavage and more mutations than just 1 copy. And then the next thing we wanted to look at is fertility. So as the midgut is a main site of blood digestion, we were interested to see if this had an effect on egg laying and hatching. So we compared the number of eggs and larvae from individual females in these samples. And although there was no significant difference in number of eggs laid, we did see a significant reduction in hatching rates in individuals that carried 2 copies of the Cas9. And so it seemed like the individuals that had this higher mutation rate in the midgut were less able to produce viable offspring. And so this is where we're at currently with the project. And the next things we're looking at are looking into some downstream genes of double sex. And so we're aiming to look at genes that could be affected mainly, and genes that particularly that are involved in blood digestion and infection. But that's where we're at so far. So thank you again for inviting me, and I'm open to any questions.
Thank you so much, Nicole. You're also good on being in time, so I never even interrupt you. Questions for Nicole, please put them in the chat. We have one question. I want to know what the real value of multiplicity of infection for polyclonal infection is. I'm not sure if this is aimed for you, Nicole.
I'm not sure. I'm not sure about that one. I'm quite new to parasites. It's not really my.
Yeah, exactly. Maybe it was lagging from before. But we have another question in the chat. So, do you think that homozygous Cas9 elevates the rate of mutagenesis?
Yeah. So from these results, it seems like it is, but I'm going to do some further analysis. In the amplicon sequencing, we didn't actually separate the heterozygous and homozygous. And so we're going to do further experiments to actually confirm that there is more mutation with the double copy of Cas9, but that's what I'm kind of presuming from these results we've got so far.
Perfect, thank you.
Thank you.
Any more questions for Nicole? I do see that after a while there come more questions, so the people that have already spoken, if you just take a look in the chat and at around the time when you spoke, and there are questions for you, so you can kind of reply in the chat. I know it's not going to be live for everyone, but at least the people will get answers. So we will move on. Next speaker is Ebenezer Ado-Ofoye from University of Ghana. And here are your slides, please.
Thank you so much, and hello everyone. First of all, I want to thank you for the opportunity given me, and then and I hope to be able to share my work I did for my master's. So, like she's right, I'm from University of Ghana, and I did my MPhil at West Africa Center for Cell Biology of Infectious Pathogen on campus. Next slide, please. Sorry, please. Can you go back just a minute? All right. So what I wanted to really understand was to be able to compare and understand how polymorphism that exists in vaccine candidate antigen impacts on T cell immune responses from T cell in low and high malaria transmission areas. Next slide, please. So, like we all know, malaria still remains a public health importance, especially in our part of Africa, which is sub-Saharan Africa. So there have been a reduction in the incidence of malaria over the years, but what you've realized that this incidence has really, this decline rate has really reduced significantly. So it tells us that if we really want to really help to eradicate malaria, then effective vaccine will be a very to be an effort to be able to complement the existing efforts. But for a vaccine to be considered as effective, we believe that that vaccine should be able to elicit immune response against diverse form of the parasite, and also in a population that is highly diverse. So to be able to contribute our quota to the search of a vaccine, I decided to really consider one of the important vaccine candidate antigen, which is the AMA1, or known as the apical membrane antigen. Yeah. So this antigen is highly polymorphic. Although it's also a vaccine candidate antibody, it's highly polymorphic. That's an unfortunate thing we know about it. And a lot of studies have been done on this particular antigen. And what we have realized that antibodies that react against this particular epitopes of these antigens are really strain specific. But I wanted to understand how T cell respond. Because we know the adaptive arm of the immune system doesn't really deal with the T cell, there's B cell. So if antibodies are strain specific, how would T cell behave when we test these peptides against T cell? So we selected two sites in Ghana, which is the Obom, which is a high malaria transmission area, and Legon, which is a low transmission area. Next slide, please. All right. So on your top left, what we want to see are these are the peptides. On the second column, these are the sequences of the peptides that we chose, and then there's the third column is the strain those sequences belong to, and the last column refers to the position of those peptides. So all these peptides were found in the N terminals, which are the parts that are really exposed to the parasite, and all of them, like I said, are found in the AMA1 is actually divided into three domains. The highly polymorphic domain is the domain 1, and that is where all these peptides really belong. All right. So when we selected our peptide, we wanted to test them against the PBMC of this subject. We've taken their samples, but we have to test them against three populations of cells, which is the whole PBMCs, and then we also have to another portion where we enrich it for mainly for CD8, and another portion where we enrich for CD4. And the purpose of that was to find the specificity of these responses, whether they were towards the CD4 or CD8 T cells. And that's how you find out your bottom left, that a representative of how the depletion were done to get our cell of choice. All right, so this, if you could see the figure with the S16 written on, is just one of the subjects. I just want to use that one as an explanation for everything that really happened. I just want to really look at it critically. So if you look at these, the bar with the blue bar represents the positive control, and the last one, which is yellow, is our negative control. So if you could realize if you look at it critically compared to the sequence of the peptide I just showed you, the one in red, green, and purple are peptides from the same peptide, but just that they belong to different strains of the parasite. You could see that this individual, who is the subject 16, made positive responses to one of the peptides belonging to the 3D7, but this corresponding variant never showed any positivity. And the same thing also is found in the one with the brown and the deep blue color. You could see that the reaction also came from there. So what we realized that there are some of the peptides that show positivity, but their corresponding variant that belong to other strains reacted negative. But if you go to other individual, the peptide that responded negative and another individual responded positive. Responded positive. So we really realized this in our studies that we realized. And then if you just come below that, and what we also wanted to also compare was these different strains of the parasite, where were they dominant? And like I said, Obom was I'm looking at figure D now. Obom was a high malaria transmission area, and then Legon was a low malaria transmission area. So you could see that subjects in Obom.
A 30-second warning.
Sure. Subjects in Obom reacted to different strains of the peptide, while those in Legon only reacted to only a single strain. And it makes sense because we know that high transmission area drives diversity. So there's a lot of diversity. No, that's the reason they mount immune response against it. Please, can you go to the next slide, please? All right, so this is what I want you to take home from my work that we did. We realized that based on the limited peptides that we tested, the number of positive in Obom was higher than those in Legon, positive peptides. And this was as expected because Obom happened to be a high transmission area. So the individuals that might be exposed to multiple strains of the peptide. So when you challenge them with this peptide, it's easy to call for immune responses to these peptides. And then like most of these peptides were directed against CD8 T cells. And it makes sense because the CD8 T cells were the portion of cell that we've deleted all the CD4. And regulatory T cells are a subset of these CD4 T cells. When you delete them, then you give the no control production, immunity. So we saw that a lot in the CD8 produced a lot of immune responses. And then position of mutation, what we realize that most of the mutation that occurs in peptide that produce positive response, we realize that the immunogenicity of those peptides were based on the position of mutation, not just the amino acid chain. We realize that some of the amino acid change to probably an amino acid that belong in the acid charged amino group. But another peptide, when it's changed to a charged amino group, still it tends from negative to becoming positive. But this is vice versa in other peptides. So what we realize is not really about the amino acid group it changed to, but the position of mutation was what really influenced the immunogenicity. So in conclusion, what we really found out that this study has really provided some evidence that just like these for the antibody, they are strain specific. For T cell also, they are really strain specific, and it's in high transmission area, you tend to find a lot of diversity in their responses compared to low transmission area. Thank you so much.
Thank you for a really nice presentation. Running a little bit short on time, but there's time for a question. So it's a very interesting work you presented, and forgive me for not being very into these kind of studies, but when you talk about the low transmission and the high transmission, do you know if the subjects have they already had infections, or do you know if they have had malaria prior to the study?
Yes, please. So we took their blood samples on a filter paper. We ran a PCR to check for their presence of parasite, and we did microscopy as well. So we realized that some of them really had parasite in their body, but those were asymptomatic. Yeah, but this presence of this parasite in their body didn't really influence the responses we were looking for, because we were just looking at memory T cells, response of memory T cells to these peptides. Yeah, so the presence of the peptide really didn't influence the results we saw. But yes, some of them really had peptides, especially those in the high transmission area. But like I expected, they were asymptomatic.
Thank you very much for that. So we will move on. And next speaker is Holly Barkwell from the U.S. Food and Drug Administration here in the United States. Please go ahead.
Hello. Can everyone hear me and see me? Okay. Hello, and thank you for joining me for this short talk, and to the organizing committee for giving me the opportunity to share this project with you today. My name is Holly Barkwell. I'm a postgraduate research fellow in the Painter Lab at the FDA, where we work with the malaria parasite, Plasmodium falciparum. Today, I will specifically be discussing one of my projects in which we aim to develop a P. falciparum reporter line that will demonstrate the effects of anti-malarial compounds on parasite life cycle progression. Next slide, please. So in anti-malarial drug discovery and development, it's important to characterize the mode of action of anti-malarial compounds and to have a greater understanding of the stage in the parasite life cycle in which the drug activity is at its most effective. A high throughput and efficient assay for determining this stage-specific activity and achieving this level of characterization would be a really valuable tool. However, currently no such assays exist. That is why this project aims to develop a P. falciparum reporter line in which transgenic parasites will differentially fluoresce as they progress through the erythrocytic development, as demonstrated in the life cycle figure on the right-hand side. This method was, the concept for this reporter line was based on a fluorescence assay known as FUCI that was published in 2008 that describes technology for promoter-driven fluorescence in live eukaryotic cells. And we have adapted this technology to develop our reporter line using whole genome promoter activity data from a P. falciparum genome-wide transcription dynamics project that Heather worked on prior to coming to the FDA, in which biosynthetic labeling and capture of mRNAs produced a large data set of nascent and stabilized transcription values throughout the IDC. Next slide, please. So from this data, we identified stage-specific promoters that we use in this assay that were selected for exhibiting peak transcriptional activity levels at specific stages with little to no overlap with other stage-specific promoter activity, as demonstrated in the individual expression value graphs for the promoters selected for ring, trophozoite, gisant, and committed gametocyte or committed ring, as well as in the graph demonstrating the peaks altogether, showing that there is little to no overlap in peak expression, with the exception of ring and committed ring. We designed vectors for these stage-specific promoters that control unique fluorescent markers under expression of these promoters, as shown in the figures below the individual expression graphs. And we are currently in the process of selecting transgenic parasites that express promoter-driven fluorescence of these vectors that we transfect. Our aim is to integrate all of these vectors into the genome to produce a single P. falciparum promoter line that will differentially express unique fluorescence and serve as indicators for blood stage developmental progression, with the hope that this can be used as a tool for high throughput compound screening in which it would be possible to evaluate the effects of novel and current anti-malarial compounds on life cycle progression in this parasite line in a high throughput format such as a 96 well plate, among other exciting possibilities. Next slide, please. So yeah, with that, I just want to thank you again for joining me for this short talk. And before I go, I just briefly want to acknowledge that we have postgraduate and postdoctoral fellowships opening up to start in fall of 2021, or sorry, actually just 2021 in general. So if you're interested in learning more about these opportunities, or if you have any questions about this project, feel free to contact Dr. Heather Painter or myself. Our contact information is on the slide.
Thank you for that very nice presentation, Holly, and super important work you're doing here. Please ask your questions in the chat. I think there are students out there in the audience that are thinking about questions right now, phrasing them, putting them in the chat box, right?
Thank you. Yeah, no worries. That was very fast.
It is very fast. Giving a 4-minute presentation goes like that. I actually had some questions from reading your abstract, but you cleared them all out for me. I do have a question for you now in the chat. Can this also be done in the mosquito cycle?
I would say yes. So there have been parasites that were developed, specifically a parasite line in which promoter-driven fluorescence in sporozoites was achieved. So I know it has been done. And what we want to do now is develop a parasite line that can express this differential promoter-driven fluorescence. So we're hoping to include every stage in one parasite line. But I know that individually, in mosquito sages, this has been done.
Perfect. Thanks for that answer. Actually, I have a small technical question, and maybe you said that before, I didn't hear, but when you're making your transgenic lines, are you like, are you injecting, or like, how are you making the?
So we transfect the plasmids into the parasite, and we do.
It's a transfection. Okay.
Yeah, and then we have not yet gotten to integration into the genome. That's hopefully what we'll be doing as we move forward with this project.
Awesome. Well, good luck with that. And there's another question for you in the chat, and we're already off schedule, so I'm going to go ahead and read it. How did you decide or choose the genes for each IDC stages?
So that was based on work that my PI did prior to coming to the FDA. She worked on a project that produced a large data set of transcriptional expression values for mRNAs throughout the IDC. So we had a set of criteria, just a short answer, that we went through in order to pick our specific promoters, with the main criteria being that they peaked in expression at the specific stage and then kind of leveled out and didn't overlap with other promoter specifics, stage specific promoters, sorry.
Very, very nice answer. So just one last question, because I think it's a really important one. Will such genetic alterations affect the fitness of the parasites? Have you seen anything about that?
That's a really great question. Yep. And the short answer is we don't know. We're still in the pilot stages of this project, so we have a lot of stuff that we have to consider as we move forward. And a lot of questions like this have come up for us already, but we don't have any answers for that yet. But thank you.
Thank you. You still have more questions to answer in the chat, and we will move on to our last speaker of the session.
Thank you.
Which is Lawrence Milan from the Messiah University here in the US. So please go ahead.
Yeah, thanks for having us. So today, two student researchers of Dr. Milan's, Mina Deep, and myself, Matthew O'Neill, will be sharing our group's shelf life study of human red blood cell cryopreservation for a laboratory culture of Plasmodium falciparum. This study was conducted by Messiah University under the guidance of Dr. Lawrence Milan and aided by Johns Hopkins' Dr. Tripathi. Next slide, please. So the Matcha Research Trust is an affiliate field research station of Johns Hopkins Bloomberg School of Public Health. And there they work to study malaria in Matcha, Zambia through culturing patient isolates of Plasmodium falciparum. However, in this area, there's a shortage of fresh and uninfected blood. Our research simply works to support theirs by developing a method to cryopreserve red blood cells that can be collected in the US, transported in liquid nitrogen, stored in Matcha's -80°C freezers, and then thawed to be used to culture P. falciparum for studies of both asexual growth and gametocyte development. We are also focused on developing a method in which we can freeze culture appropriate volumes of red blood cells, as opposed to much larger quantities. Next slide, please.
So our study uses O positive and A positive blood that's anticoagulated with ACDA. And we use two cryopreservation methods, hydroxyethyl starch and polyvinyl alcohol, HESPDA, and glycerol serum. And they both work to protect red blood cells from ice crystals during the freezing and thawing process. We then store it in 2 milliliter preservation tubes at -80°C for various amounts of time, ranging from a couple of days to a couple of months. And then we've obtained from Hopkins, and we culture it through serial propagation of asynchronous cultures. And so our culture starts at 0.5 parasitemia, and over a 96-hour period, we monitor it up until it reaches 5 to 10% parasitemia. We then create another subculture where we dilute it back to 0.5 parasitemia, and that will represent one serial passage. And then throughout the experiment, we'll create gametocyte cultures on top of that. And then each of these cultures are incubated in a candle jar desiccator at 37 degrees Celsius and cultured in RPMI 1640 media that's supplemented with the combination of human AB plus serum and Albumax-2 alongside the gentamicin. We then visualize the parasitemia with Ginza stain smears. Next slide, please. So the yellow line represents the HES-PVA frozen blood, and our data shows that it kind of stalled at the third serial passage. Although we don't have that much evidence, we do have some theories as to why this may occur. And then on top of that, we were able to establish gametocyte cultures during the first serial passage, but we were not able to pass that first serial passage. For the glycerol serum frozen blood, which can be seen by the red line, it continued to culture the malaria strain beyond the 8th serial passage. Additionally, we were able to establish a gametocyte culture during the first serial passage, and we are currently establishing more gametocyte cultures beyond the first serial passage. Additionally, in the future, we hope to establish a shelf life study with glycerol frozen blood that's been frozen for around 6 months to a year. Some of the drawbacks of using the glycerol frozen blood is that it preserves fewer red blood cells during the freezing process, and it requires serum to freeze it, so it's more cost effective than the HES-PVA frozen blood. And so at this time, I'd just like to thank Messiah University for funding and making this research possible. Thank you.
Thank you for that presentation and for setting a good example wearing your mask when you're that close to each other. If there are any questions, please put them in the chat box. It sounds like a really important work you've been doing to help them and closer to the field. So we have a question in the chat. What was the morphology of the parasites preserved? Have you looked at that?
Yeah, we're preserving the red blood cells, but the parasite culture seems to be going through its normal stages in our culture.
Okay, and another question is, do you see any visual differences in the RBS from different conditions?
So for the most part, it looks the same between the two conditions, but like due to complications such as like bacterial contaminations, sometimes the red blood cells did look different, but I think we believe that it's due to outside reasons rather than the actual freezing techniques themselves.
We exclude the contaminated.
Yeah. And we excluded the contaminated cultures within this data.
Okay, let's do one last question before ending. So have you thought about measuring the metabolites in the RCB before and after freezing, or just the amount of glucose?
Yes, we have, and we've also thought about supplementing the red blood cells to try to find that key missing metabolite that we believe might be there. But we just haven't gotten to those studies yet, so hopefully we'll be able to continue with that in the future.
Okay, so I'm gonna have to, there are some more questions in the chat, you can still look at them, but from, since the time is ticking, I'm gonna thank all the speakers for this session. It's been short but intense and great to hear so many in this short hour. So on behalf of the organizers, I would like to thank all the attendees also for the Future of Malaria Research Symposium. This event is sponsored by the Johns Hopkins Malaria Research Institute and would not be possible without the leadership and support of Dr. Peter Auger that you met this morning, and also Genevieve Williams. We're also grateful for the technical support from Sean Rogers, Tyler Hill, and Anna Coyne. Without them, this day would not have been possible, but I think we've all become a little bit more Zoom experts this year. 2020 has been a challenging year. The silver lining of this pandemic is the ability to have scientists from across the globe come together for this symposium. I'm so glad to see speakers from all the different continents and to share your research. We're grateful to all the speakers that participated today, and I hope that you will join us next year for the World Malaria Day, which is taking place on April 23rd. Thank you all. This is the end from my end.